All right, I should probably introduce our today's genetics colloquium speaker. It's my pleasure to introduce my colleague and friend, Maitreya Dunham, professor of genome sciences from the other UW Seattle. Maitreya got her BS in biology from MIT and her PhD in genetics from Stanford, training under David Botstein. So, pretty good. She did her postdoctoral work as a Lewis Sigler Fellow at Princeton. And since that time, she has won a number of awards and accolades for her independent work. One of the most recent being named a Howard Hughes Medical Institute faculty scholar. Her lab develops and uses cutting-edge genomics to explore fundamental and practical questions relevant to genome evolution and genetic variation in yeast in humans. I met Maitreya through a fun collaborative project that we have been trying to rekindle, which is why she's been here for a few extra days. And because she's a great service-minded citizen scientist, one of my favorite things about her. And she ran the Cold Spring Harbor Laboratory yeast course for a number of years. And for some reason, thought I was worth inviting out for a talk to her 2016 class. And we had an absolute blast. I've not ever been a person who likes camping, but for the first time in my life, I felt like I was science camping. And what was really fun was, at the time, this silly game was popular. It was called Pokemon Go. And I was really into it because I was playing it with my 13-year-old. And I was so happy to learn that all the students were interested in it. And so was Maitreya. And we were totally into Pokemon Go. So in between asking really cool questions, in between asking really cool science questions, we also discussed powering up our Pokemons. In fact, I once accidentally asked that question during one of the science sessions because I just was fascinated with this particular one. And it was just cool. And she was able to do like intense, fantastic scientist. It was really science. It was really cutting edge, but she had a sense of humor and whimsy about it. And that was basically my favorite type of person, right? Great scientist, but a totally cool person you want to hang with. And I'm sure we'll enjoy her talk today. Trish.
Oh, that is so nice of you, Catherine. Thanks so much for the introduction. And, we've been working on evolution together, I guess, in multiple venues for quite a while. So today I thought I would talk about two projects that we have in the lab where we're moving experimental evolution, which we've been doing for a very long time, out of the confines and the controlled space of the lab and into wilder environments. And so experimental evolution is a really powerful technique where we can watch evolution happen in real time in the lab and use all the powerful tools of sequencing whole genomes to be able to kind of follow what happens in the genome. And so, we can subject the yeast to some new environment, allow it to invent new capabilities, adapt to a stressful condition, and then, see what happens. And this style of experiment in microbes only takes a few months before you can see some phenotypic change on a population scale. And the goal, of course, is to be able to understand better the molecular basis of genome evolution, but also, it's a real great model, I think, for more complex more complex evolutionary scenarios like cancer progression or evolution of antibiotic resistance, in addition to being kind of inherently interesting. And so the questions that we had that I'll talk about today are, can we use this also as, watching evolution is powerful? Could watching evolution be a powerful teaching tool to understand evolution in that context? And are there ways that we can kind of make things more complex and enter the real world as opposed to these kind of toy evolutions that we've been doing in the lab? So I'll start with this teaching tool one. So this project actually started when this guy here, Ryan, emailed me out of the blue and asked if yeast experimental evolution was a thing. He had read about Rich Lenski's long-term evolution experiment with bacteria, but he taught AP biology in an American high school where eukaryotes are the main kind of learning objectives that are the whole course is organized around. And so he thought that maybe what he could do is to make his teaching lab more interactive, and also to teach students more about what science actually is, that it's a process and it's still going on, as opposed to just a book of things to memorize, that he wanted to engage with students in actual research. And so he said, well, I Googled yeast experimental evolution and you were the top hit. Would you be interested in working with me? And the funny thing about this also is that my top hit, I did this experiment in incognito mode, and a top hit is a book chapter that I think only I have ever cited, but it has like amazing SEO rank for some unknown reason. So anyway, writing a book chapter is a good thing. So the other piece of this puzzle was my postdoc, Bryce Taylor, who had started in the lab a little while ago and had always been interested in outreach and had as a goal starting his own lab at a primarily undergraduate institution. And so the match was made. And what I'll tell you today started with Ryan, who works at an all-girls private school that is, let's just say, highly resourced in Southern California, but we've now extended it into a number of other schools, public schools, and with other partners as well. And Bryce is now actually at Loras College, where he has started a new franchise of this model, and we're continuing to develop it together. Okay, so we're calling it YEVO, and the students evolve yeast in their classrooms. And we have a few different modules that kind of allow some flexibility for the teachers. So they evolve the yeast and we sequence it. They do a genome analysis module that we're trying to bulk up so that they get some exposure to bioinformatics. And then they think about fitness by virtue of watching how things grow and by competition assays. And then they get to start thinking about some more kind of complicated ideas in evolutionary biology, like trade-offs, which kind of get them deep thinking. Okay, so what do we need in order to make this work? Well, obviously we need our platform. We need yeast strains, selective pressure, fitness measurements, and we are going to sequence their genomes at the end. So I'll step through each of these. So in my lab, our favorite way of doing evolution experiments, where we can control as many things about the process as possible, is with continuous cultures. So this is a setup of our chemostats, where we like to do evolution experiments. And just one look at these and you can tell high school is not an appropriate place to set up this. I mean, even the undergrads in my lab, it takes a while to get them fully trained on this gadget. So what we needed was something simpler. And so what we developed was a serial transfer method, where we wanted to also decrease the possibilities of contamination over time. And so what we ended up with is individually wrapped sterile cotton swabs that the students use to dip into their culture every day and transfer into the next day's culture. And so by doing this, it's a very simple transfer. It doesn't take up much class time. And the students can actually do replicates as well. And this protocol here was drawn by a graduate student in the department, Sai, who is a fantastic illustrator, but made this for us when we made this into a kit for students to do evolution experiments at home, and they needed kind of a graphical protocol. The strains that we use are, except for the home one where we use just baker's yeast, we use these beautiful strains that we got from both Jeff Buca's lab and also that the students at the yeast course that Catherine mentioned made. So some of these express the beta carotene biosynthesis pathway. We call these the carrot yeast, and we get all sorts of colors from yellow to orange, depending on the strength of the promoters we use. Some of them have genes from like coral or bacteria that make these beautiful pigments. And so each team gets the color, and that's their ancestor. And that allows us to monitor for contamination. We have occasionally had a student inoculate the wrong tube, and then the next day they come in and it's muddy colored instead of whatever color it should be. And so then we know to go back in time to the fridge and start over again. But also it's going to allow us to do head-to-head competitions, which I'll get to later. The other thing that was kind of unanticipated is it gives the students almost a team identity. So it's their strains that they're evolving. And actually, when we did our surveys of the students to find out what elements of the lab they liked or didn't like, one of the complaints we got was the students who got this dark gray strain here were very unhappy. And so we're trying to, we clearly need more like emo goth students in our class for one thing, but second, we're going to try to rebrand it this year as silver and see if that helps. So that was my undergraduate Naomi's idea. So, okay, so what selective condition to use? So as a first pass at this, we wanted to find something that was easy, that we knew would work, and that also had some relevance to students' kind of daily experience of the world. And so antifungal resistance is an easy analogy to antibiotic resistance, which is often in the news, and which kind of is interesting, I think, from multiple perspectives, including public health. And also, you know, most people in the world have taken an antibiotic of some sort or another. And so Bryce and Ryan went down to the Ave next to UW to the drugstore, and they bought every over-the-counter antifungal that they sold, which the cashier, I'm told, took their money very gingerly. They tried all of them against lab yeast. And it turned out this one here, Fungicure, which has the active ingredient of clotrimazole, and a 1 in 50,000 dilution, and I actually need to edit this slide. Kills is the wrong word. So these are fungistatic drugs, so they prevent growth of the yeast. Okay, so what's nice about this is that azoles are a really important class of antifungals. In fact, a huge problem in antifungals is that we have so few categories of antifungals to use. It has a known mechanism of action where it interferes with yeast ergosterol synthesis. But there are also a lot of known resistance mutations. So we could have an element of possible discovery, but also have some grounding in literature. We gave the students the choice of what drug dosages to use. So this was one part that really was very effective, because the students had ownership of their experiments in a way that led to, you know, we gave them room to fail, actually. So we occasionally had extinction events where they would go too far too fast and all their yeast would die. And this also came up on our evaluations, that makes you sad when all your yeast die. But we kept backups in the fridge so they could go back in time again and restart. But they got to think about, like, oh, I've seen improvement, maybe it's time to increase the dose. And they all had different strategies. And since they worked in teams, there was an element of kind of cooperation, and they had multiple replicates. So they could see that things were not progressing the same in all their populations. And so it's a way of starting to appreciate that element of stochasticity in evolution. Okay, so the first data sets that we collected were with Ryan, and then also Paul Rowley has been a great collaborator on this. He's at the University of Idaho and recruited 2 Moscow, Idaho teachers in on the project as well. And so they did evolution experiments, and we had to make some modifications depending on the school. So at Westridge, they had longer periods of class so they could spend a little more time on things. At Moscow, we only did back dilutions once a week. And so there's some but what we found was that things were pretty robust to this. So we also have 2 years worth of students in both classes. The pandemic screwed everything up, but we're back on track now. And what we found was that all of the classrooms were able to evolve appreciable levels of drug resistance that we could measure and that they could see. And so, so here is just the ancestor strain, only 2 micromolar. And then these are histograms of the evolved strains that we got from each of these classes. So we sequenced 99 of these. This is actually now the highest replicated evolution experiment I've ever done in my lab, thanks to these student collaborators. And the sequencing we did in my lab, but also Fritz Roth, I was telling him about this project, and he says, you know what, I co-teach a class with Atina Cole, and they sequence things in the class, and could we sequence your strains? And I was like, that's perfect. So they're going to do this again for us this year, which has been great, because the sequencing is the most expensive part of the whole thing, in addition to the people, of course. And so we saw all sorts of different mutations in these strains, and all of the evolved strains had at least one obvious candidate that was known in the literature to cause drug resistance. But it was fun to teach the students also about different types of mutations. So we saw copy number changes. We saw changes in organellar genomes, like mitochondrial genomes. We saw transposon insertion changes, and of course, point mutations. So here are some of the copy number changes that we saw. So ERG11 here is the drug target of the azole family. And here you can see we got a bunch of amplifications that include a segment of including this gene. And in this case, we got 38 strains that amplified the whole chromosome. And so it was kind of fun for the students to see this, and the analogies to cancer and fungal pathogens. We could kind of connect those dots for them. We're not entirely sure what the target gene is in this region, because you can see that all of these don't totally overlap. Our favorite candidate gene here that's a regulator. And then we found a bunch of strains that amplified a drug pump. So this one's especially fun because well, actually, this is not the best example of it. I'll show it in a minute. So the best example are PDR1 and 3. So these are point mutations that we saw in nearly all of the strains. And so one great thing is the students, like we give them their mutation list, and then they start looking up these genes, and they type PDR1 into Google or into SGD, and it stands for pleiotropic drug resistance 1. And they go, oh my God, it worked. So sometimes, they really, it was nice to see that there were these very obvious, these are paralogs that are transcription factors that regulate a lot of the ABC drug transporters and other response to drugs. So it's no surprise that these are likely gain of function mutations that activate downstream. And so one complication of the first round that we did of this was that how to involve the students in the bioinformatics was really challenging. We were doing the sequencing and the analysis, and we have a kind of kludgy Frankenstein's monster of an analysis pipeline that I see some people nodding in the audience who also but it works. And so, but it's not friendly for anyone pretty much outside my lab even to run. And so the students, so they want to get into programming. They want to understand the analysis. And so we've been really trying to work harder at this. And so one advance this year was we had a hackathon in our department, and one of the teams took this on as a challenge. And so they made this great R Shiny app browser where the students can choose what classroom they're in, look at what year they are and what selection pressure they have. And it shows not just their mutations, but all the other ones in the class. So here they can see, oh, we got a whole bunch of mutations in PDR1, or here we got a whole bunch of mutations in PDR3. And then they can also compare to other classrooms. So we're trying to give kind of a team, like you're part of a bigger project aspect to this, but they can also compare notes with other classes. So we're still working on this Shiny app, but it's been fun to see, like we're going to beta test it this year and see how the students like it. One of the other types of mutations we got are petites. So yeast can survive without their mitochondrial DNA. And if they lose their mitochondrial DNA, they also lose the ability to grow on a non-fermentable carbon source. So you can see that here when we grow these evolved strains, here are our ancestors. They all grow on glucose and they all grow on glycerol, but many of the evolved strains have lost the ability to grow on glycerol. You can also just see how much fun it is to work with these strains. Like they look like candy dots, right? I mean, it's just really pretty. So even a spotting assay is fun. We actually are not 100% sure what mitochondrial activity has to do totally with drug resistance, but we did find some hints in the literature that in Candida species, there are links between mitochondrial activity and activation of PDR family of genes. So we're on to something, we're not entirely sure what, but here is just kind of a summary of a bunch of the different types of mutations that we got. And also an example of how by doing something 99 times, you can start to see patterns emerge. And so here, for example, if you look down the column here, nearly every strain has a mutation in either PDR1 or PDR3, but never both. And so that was pretty interesting. And we actually made the double mutant and it doesn't have a benefit over the single mutant. So this is an example where they're non-additive because they're redundant, right? We also saw some interesting patterns of particular mutations that occurred on a background of an ERG25 mutation. So the ERG25 mutation appears to have happened first, and then these mutations were layered on it, which we kind of inferred from some timing sequencing that we did, where we have both early and late time points for some of these experiments. Reconstructing the double mutants of some of these has led to some kind of confusing results. So we're still in the process of following this up to see if these patterns actually indicate something about how beneficial mutations combine. So overall, we found all sorts of different mutations. It's been super fun to just sift through these. The students have to write a little essay about their favorite gene and what it might or might not have to do with drug resistance, and they get really into it. A couple of the students spent a day off once working with Ryan looking up yeast genes. And at the end of the day, one of the students said, I think this might be what a scientist does. And I was like, oh my God, it's like, my heart grew 3 sizes. And it's like, it's just been pure joy working with this. The students get so into it and everything they find is new and fun. And actually it's new and fun for us too, because they actually found some probably new things. So all the blue ones here and the green ones are genes that had been previously implicated in antifungal resistance. But we found multiple mutations in independent populations for several genes that were, we couldn't find any connection to antifungal resistance. And so one of these, CSF1, we tried to follow up. All we did was reconstruct the mutation using CRISPR, and we're now trying to actually build this into a side lab if teachers want to teach a CRISPR lab. But what you do is you just can compete these against each other, either in a condition with no drug or in a condition with the drug. And after you propagate it for a little while, we just use Sanger sequencing, actually, in this case, to tell if one's winning or another. You can go in and look at the height of the little trace here, and you can tell really well, actually. And so we were trying to also come up with something relatively simple that classrooms could potentially do. And so in the no drug setting, these all kind of are equally frequent at the beginning and at the end. But in the FungiCure condition, we see the CSF1 mutation sweep. So CSF1 does appear to be an authentic antifungal target and is related to something new. It has some relationship to mitochondrial biology, again, that we're not totally sure about, but Bryce is going to follow this up in his new lab. He's got some new master's students who are excited about it. So, these competition assays are a great way of understanding fitness. And so the students get to get in on this too. And so because these colors, and we could have done this experiment that way, we could have made the wild type here a different color, but here we have, the students can have different evolved strains and then ask who evolved the best yeast. And they can just figure this out by simply plating and counting colonies of these different colors. And so we get really into this. The students love it. We call it yeast madness and set up a whole bracket. One of my schools in Seattle, they actually handed out brackets to the whole school, not just class. And the whole school was following yeast madness. And they're like, oh, I don't know. I think the purple strain's going all the way. It had that PDR1 mutation. I don't know. It's just been great. And so, and whoever wins, they get so into who's going to win, right? So that was one of our other negative complaints on our course evaluations, is that, a bracket is only going to have one winner. And so everyone who is a loser is happy about it. So, but, try again next year. And so, and the other thing that we found out was by talking to the teachers that even though this was really fun and the students got to think about what does competitive fitness mean, and actually this is where the trade-offs come in. So those petite mutants, they're called petite because they make tiny colonies on glucose plates. So they do the competition, they find there are a whole bunch of petite colonies and a small number of the competitor, but the competitor colonies are huge and the petite colonies are tiny, and they're like, but it's the winner. And then they think very hard. And so that's been kind of fun. They get to see elements of fitness that mean different things in different environments. But the other thing was that the bracket where we had rounds took too much time in the lab. And so we have now, my student Naomi took this plate, and this is the melee battle option, where it turns out you can differentiate 6 different colors pretty well. And the teacher who piloted this year, Rebecca Brewer, who's in Michigan, she sent us some photos of her students counting these plates, where they had taken photos with their cell phones and then zoomed in on them and had used like a pen app to mark the colonies. And I was like, that's really smart. So they have a good time with it. Okay, so this is really adaptable. So with Rebecca, that person I just mentioned, we've actually been doing caffeine resistance with her, which we started doing during the pandemic when we wanted to do home evolutions, and I knew somebody's dog was going to eat their experiment, and then I was going to get a phone call, right? So we wanted something really safe, and caffeine is a TOR inhibitor, and we've gotten really cool mutations from this. We've also been doing what we call the sludge evolutions with a biotechnology class outside of Seattle, and that's with this wood waste hydrolysate that we get from the forestry department at UW. One of my undergraduates devised a way of doing invasive growth where the cells can kind of grow into a substrate like this. And so she just takes a plug of this and propagates it, and was able to evolve more invasive yeasts in just a few passages. And so we're now trying to develop that because this is a really nice visible phenotype, right? We've done industrial strains and baking strains for what we use for the home evolutions, which the beta tests were done by my lab members when we all were stuck at home early in the pandemic. And student side projects. We've had a few students do science fair projects related to this, and we just put them all up at our website too. We've got a few student projects up there. And then so the hope is that now that this is pretty fully developed, that we can start more franchises, that it took Bryce a couple of years to develop this and for us to work out the kinks, and we're still developing it, obviously, but it works pretty well now. And so we think it's ready to ship, so that if other people would like to adopt it, create their own selection pressures, you can get your own high school workforce to be doing experiments for you, and you can get all this great satisfaction of working with all these schools and students. So we have a website that also has that mutation viewer there, and a Twitter feed, and the paper about the mutations just came out actually a couple weeks ago in G3, including that new gene that we think is relevant. And we have a preprint up that's about the pedagogy that includes some of these evaluations. And getting into education research has been its own learning curve that I'm not totally sure we've figured out yet. And so we were able to partner with an education researcher, Alexa Warwick, for that, which has been really great fun. And then this caffeine resistance experiment, we also have made a lot of progress on, and we're trying out a different type of publication for this called a micro publication, where the undergraduate who was working with the classroom, she actually did a ton of research into all the different mutations that she was seeing. And so she was able to write up this one-figure paper on her own with some input from us. But I think it's a really great venue for the undergraduates to get involved and have their own work come out of it. Okay, so that's the first project. So that's trying to simplify evolution so that where high school students can do it and then use those results for science and also for outreach. The other project that I want to talk about is kind of a way of complicating experimental evolution in some sense. But these really simple evolution experiments we do in the lab are great, and we actually still don't understand all the mutations that we get out of them. So we haven't solved evolution in that sense. But kind of getting out into more complex environments, more ecologically realistic environments for yeast is something that, it would be great to be able to do. And so it turned out that we didn't have to develop this ourselves because it was already happening in breweries all over the world every day. And so I found out about this thanks to a talk series that the graduate students run at University of Washington on what can I do with my PhD in the biosciences. And so they bring in speakers that have awesome jobs where they use their PhD, and a variety of jobs, right? So that you get kind of an idea of different things you could do. And so one day I walked by a poster that was a brewer who has a brewery called Postdoc Brewing. And he was a PhD graduate from the program and was coming back to give a career talk. And I contacted him and was like, hey, I'd be interested in talking to you about what you do. And we talked about repitching, and he told me about this process where brewers reuse yeast over and over again. And most places only reuse their yeast a limited number of times. And that's because things change, the way the yeast behaves change, but maybe also the yeast change because of evolution. Maybe there are mutations like this red one here that go up in frequency and change the genotype of the yeast, which changes the kind of extended phenotype of the yeast, which is beer. And so this looked remarkably like experimental evolution to me. And so we struck up a collaboration to collect time courses. And so here's Tom, and you can see here is industry survey of a small number of breweries, but 12 to 15 is like the top of the graph. So Tom actually is off the chart. So the first data set we collected with him was 26 cycles of beer, which took about 6 months. And I think part of this might be that Tom perhaps has better sterile technique than some brewers, perhaps, given his scientist background. But also breweries are like shoestring operations. Like, they are not like reeling in the money. And so yeast costs money to buy from the yeast supplier. And so he wanted to reuse his yeast as long as possible to not have to repurchase it so often. And so, you can imagine if you're starting with new yeast every 5 to 8 cycles, then, only having to do it every 26 cycles, not bad, right? So he's just right across the lake from us. And one of the people who works at his brewery lives pretty close to UW. So he would drop by with yeast samples whenever they'd completed a cycle. And so the idea now is that we have these samples and we can use, again, the same technique we use for everything, just whole genome sequencing to find out, are there differences? The one difference between the way we're doing the sequencing for this part and for the part 1 was that we're sequencing clones on part 1. The students did a good job, but we weren't confident that the populations would be totally pure. and not contaminated with something or another. And so we wanted to isolate clones, recheck that they actually had the phenotype we were interested in, and then sequence them. But for these, we just sequenced direct from the population. So these are now mixed cultures. And to be honest, I wasn't totally sure this was going to work, because while I've shown it as now this is like a large portion of the population, this red one, it could easily be that there are a bunch of subpopulations that all have kind of clonal interference and never individually rise to a detectable frequency. Or this strain could be so well adapted to brewing conditions, since it's been selected for hundreds of years to do that, that there's really not a lot of evolution left to happen because it's very well adapted. So luckily, Chris Large, a graduate student in my lab, was willing to take this on. And Chris is both an excellent evolutionary geneticist and also a home brewer. So it ended up being a great partnership. And he worked closely with a technician in my lab, Noah Fredstrom, who also is a home brewer. And so Noah actually came up with a recipe for what we jokingly call science beer that we use to make sterile wort and do experiments in the lab. But Chris did all of the genomics work that I'm about to show. Okay, so before I show you the results, I want to introduce you to ale yeast. Ale yeast is Saccharomyces cerevisiae, and it is likely the result of some sort of mating event or fusion event from hundreds of years ago that created a strain that has 4 copies of each chromosome and 4 different haplotypes. So these 2 progenitor strains that Justin Fay and Amy Dudley kind of hypothesized were the ancient parents of this, both were kind of heterozygous when they started. And so we've got a very complicated starting genotype here with a lot of standing genetic variation within the strain. And then this gets propagated for however many generations in breweries and other environments. And over that period of time, some chromosomes maintain their ancestral diversity and copy number, but we also see some chromosomes that change copy number. We got some chromosomes that get recombinations, mitotic recombinations, I should say, because meiosis is off the table for most of these strains. And so the question was, if we now took this complicated strain and evolved it further, what would we see? One thing that is difficult about these strains is, and this is just one cartoon here, there are hundreds of brewing strains that have now been sequenced by a variety of groups, including ours, and they have different constellations of chromosome rearrangements, number changes, and things like that. And while there are a few loci, some of which Chris has been involved in working on, that are known to be related to the domestication of these strains, the relevance of the large number of genetic differences among them all is really difficult to parcel out from such complicated genomes. So we thought by following their short-term evolution that was from de novo mutation, we might be able to use the same kind of shortcut that we were using for experimental evolution in the lab. Okay, so what did we find? So this is just now looking at read depth of one chromosome, chromosome 5, and each of these is a different time point up to pitch 26. And so what you can see is there's increased read depth and increased copy number by the end here. And actually what this corresponds to is 40% of the final population had an extra copy of this chromosome, 40%. So my prediction or worry that maybe we wouldn't be able to see anything at the population level turned out to not be true. And we can tell from, so remember I mentioned that there are different haplotypes. We can actually tell that this extra chromosome copy occurred independently multiple times within the population, because we see examples of each one of the haplotypes being present in extra copies in different lineages. So that's kind of interesting. So it's really a cohort of mutants that are going up in frequency here. We have some ideas of candidate genes on this chromosome, but we haven't followed up any of them. So I decided to not talk about them today for the sake of time. The second type of change that we saw was a little bit more complicated to detect. And so we're going to, it's copy number neutral. So we don't actually see any change in copy number here, but we see a change in what haplotypes are available. And so here, for example, you could imagine 2 scenarios where you either have all the chromosomes are the same, meaning that if you look at the non-reference strain allele, it could either be at, fixed or none of them. Or if you had a 3-to-1 ratio here, you can get different frequencies, right? And you can imagine that could happen in different ways, right? You could have all sorts of different scenarios here where you have, and 1 to 3, I should say, is some of these are ambiguous, right? So like you could have a new mutation that occurred only on this haplotype, or that is left over from an ancestral haplotype, or it could be on this other haplotype. And so one thing that helps us is that we can look along the whole chromosome length, and that's what all the dots are supposed to represent. And so if we see a cohort of variants all changing together, we can kind of infer something about that haplotype's copy number. And because these are tetraploids with 4 different possible haplotypes, these get complicated quick, especially if you start to layer aneuploidy on top of it. And so, we can get all sorts of different ratios depending on how you parcel out the variants that tag each haplotype. Okay, so what do these data look like? So this is our starting strain. So the starting strain has already, you can see, had something happen that has changed the haplotype content on the 2 different arms of the chromosome. And so here we can see 3 quarters, 1/2, and 1/4. And so what we think is going on here is that we have 2 haplotypes at 1 copy and 2 at 2 copies, and that 2 of them are more similar to each other than to the 3rd. And so probably the 2 that came in together from the original parent strain. And on the other arm of the chromosome, we've gotten rid of one of these minority haplotypes and replaced it with an extra copy of the majority one. And so over time, what you can see is that this arm of the chromosome doesn't really change, but this arm of the chromosome, we see kind of this sloping pattern. And what this indicates is an increase in the frequency of the minority haplotype and a decrease in the frequency of the majority haplotype. So it's going from 3-to-1 and 1-to-3 to more like 50-50. And we were able to prove this by sequencing clones. And we're also, and you might also be like, well, if it's this, then why don't we see a step function? And that's because this event also occurred in multiple subpopulations here. So by sequencing clones, we were able to find clones with different mitotic recombination breakpoints that tiled over this region. And so all of these have in common the subtelomeric end here, but different decreasing frequencies in the population carried larger and larger and larger ones. So that's why we're seeing this kind of sloping pattern. If we use kind of the end here, and I'll get to this complicated pattern later. If we get to the end here, we can estimate that, again, approximately 40% of the population carries one of these recombination events. And in this particular experiment, it's a different 40% than the 40% that have the extra chromosome. That's not true in other experiments that we've done. But so in this case, we've now found 80% of the population is mutants. So I've started, when I talk to new brewers, giving them the caveat that I'm going to tell you that you have mutant beer. And if you think your customers will not be okay with mutant beer, then we can, blind your identity, or you cannot do this collaboration, but just want you to know. But I mean, we're all honestly drinking mutant beer already, right? So Chris is a good geneticist, and he was like, well, that was 6 months of brewing and, a good few months of his work to analyze the data to do one replicate. But, were we lucky? Does Tom do something weird? Was this abnormal? And so Tom agreed to collect a second time course for us, went out to pitch 29 this time. And to my great astonishment, we got the exact same mutations, increasing in frequency over time in the second time course. In fact, we also got a third mutation that I'm not tracking here, but a chromosome 15 mitotic recombination. It looks quite similar to the kind of pattern that I showed for the chromosome 8 one. And then we thought, well, let's go do some other breweries. And so Chris had joined the American Society for Brewing Chemists and was on their message board asking if there would be people interested in this. And so we were able to recruit a few new collaborators to this, one in Seattle. It turned out the head yeast biologist there had been on her quals committee and that was what she was doing. She now works at a biotech company, but it was really great to collaborate with her. And we were calling them Brewery X for a long time because she took the kind of ask forgiveness, not permission strategy, because they're now owned by InBev. And so we weren't totally sure how they'd like us sequencing things, but they were fine with it once we told them about it. And so she's a co-author on the manuscript now. And so interestingly, the strain that they start with is a little bit different and already had the chromosome copy number chromosome 5 that we saw occur as a mutation in these populations. However, we also saw this chromosome 8 mitotic recombination happen over just the course of the repitching. And we kind of see similar things here where we have some pre-existing events, but others that we see again happen novo. And this was really interesting to us because at the other thing that Chris was doing on the message boards was collecting every type of brewing strain related to this family of brewing strains that we could find. So this is part of a family called the Chico variety. It was popularized by Sierra Nevada, which is based in Chico, California. And it's often called American ale, or all the yeast companies have a different name for it. But it's a very frequently used kind of all-purpose ale brewing strain. And so we'd sequenced a whole bunch of them collected from different companies and also different house strains that different breweries had adapted for their own use. And so what we found was that some of the mutations that we were watching happen de novo had actually happened across this tree also. So going from 3 to 4 copies of chromosome 5, the blue strains here all have 4 copies. And then the chromosome 8 mitotic recombination has actually happened in this entire lineage here. So that's kind of interesting. So what about this chromosome 8 region? I just want to talk a little bit about the genes that are there and why we think it might have been beneficial. And so Chris was really staring at this data, trying to understand what's so special about chromosome 8. And he happened to notice that some of the strains, the postdoc brewing strains in particular, had a point mutation in a gene just on one haplotype here. And that gene was BAT1. And BAT1 is a very suspicious gene to find a mutation in because it's involved in branched amino acid biosynthesis. And that is involved in creation of a bunch of alcohols that are not ethanol, but are other types of alcohols created and can create different flavors. And so we were at a conference a few years ago, and Chris was talking about this and met the world expert in BAT1 biology, Hiroshi Takagi, who agreed to collaborate with us and create this allele in his test strains, which are lab strains, and test if it had a functional difference. And so here are the levels of these alcohols, again, tested in a lab strain with the wild type sequence, with the gene deleted entirely, and now with this minority allele in the brewing strain. And sure enough, this sequence appears to be a loss of function allele. And it had, so this is a mutation that could have had an effect on flavor, but it also might have a growth phenotype. So turns out this gene is also involved in osmolarity tolerance. And so here's the wild type here. It grows well, even up to very high concentrations of glucose. And the deletion allele and the brewing strain substitution also have a growth defect at high osmolarity. And so wort, has a lot of sugar content. And so it could be that the strain with one of these alleles has a defect in growing at the initial stages of a fermentation when it first hits that high sugar environment. However, we did find that some of the strains did not have this BAT1 allele. And so in particular, the Elysian strain did not. So the BAT1 allele came in just this clade. And so we wanted to look further at the genes on that chromosome. And so this is where we needed to end up going to whole genome sequencing, because we kind of noticed that our sequencing data started looking weird once we got down to the telomeres, and telomeres are sites of frequent recombination in different yeast strains. And so with Joseph Schescher's lab, we were able to do nanopore sequencing of these strains. And we found that sure enough, the different haplotypes don't just differ according to which allele of BAT1 they carry, but also they differ by what telomeres they contain. So this minority haplotype had actually a telomere from chromosome 1, and even the chromosome 8 telomere wasn't actually there. It was actually chromosome 9 telomere in this particular strain. So kind of interesting. And so this gene here is very suspicious. And so it's called LG-FLO1, and the LG stands for lager. So that was like, oh, we're onto something here. So that's because this was a gene that was found in a lager strain originally. And what this gene does is it controls flocculation. So, yeast in the lab has had this bred out of it because we need single colonies to come from single cells. And also dealing with clumps is just annoying. But many yeast in the wild and also industrial strains will clump together. And that's generated by these protein-protein, manoprotein interactions on the surfaces of the cells. And so what had happened here was by getting this recombination event, we got a twofer. We got a replacement of the bad BAT1 allele. So the strain no longer has this baggage of the bad BAT1 allele. And now it's got an extra copy of LG-FLO1. And we'd actually missed this because we tested flocculation to see if that was a phenotype that mattered here, and hadn't seen a difference until Noah created this lab wort recipe, and it turns out LG-FLO1 is maltose dependent. So this is a maltose dependent flocculation phenotype that this gene is related to. So we redid all of our experiments in maltose, and then here's what we got. So here are two isolates of the ancestor strain. They have kind of medium flocculation. And the way we measure this is you literally fill a tube with yeast, you let it sit there for a few minutes, you pipette off the very top, and then you check the spec to see what the OD is of that. And so if the yeast all sink to the bottom, then you see very low optical density. And if the yeast are staying aloft, then you can see a kind of medium optical density. And so a positive control, they all sink right away in the 6 minutes. Our evolved strains that did not have this chromosome rearrangement that created an extra copy of this segment all had pretty similar flocculation phenotypes to the ancestor. So the strain with the extra copy of chromosome 5, or we did find a couple of strains that had a mitotic recombination on chromosome 8, not between the dark blue and the light blue haplotype, but between two light blue haplotypes. So it still got rid of the bad BAT1 allele, but it didn't do so by also amplifying this other allele. Most strains, however, amplified the other allele, and these all that had now 2 copies of this telomere had increased flocculation. So we think that this partially explains why this mutation has been so popular in these particular evolution experiments or repitching experiments. And so one question is, well, what's changed? You know, these strains are brewing strains. They've been used for brewing for eons, you know. And so one thing that we think has changed is that kind of yeast evolution is kind of frozen in time due to modern freezers, that we have yeast stocks that are, you know, set in ice where you can go back and get them anytime and get the same strain, right? They had, you know, previously been propagated. And one way that you propagate yeast is you can either collect from, you know, you have a giant stainless steel fermenter with a cone in the bottom. And so what brewers usually do is they collect from the cone because it's easy to just wait for the yeast to settle out. But top skimming is historically how ale yeasts were repitched. And so this is a picture Chris found in a brewing history book from a brewery called Ballantine Brewing, which is actually where the Chico strains originally probably came from. And so this is all yeast foam that's being skimmed off to be used in the next batch. And so what we think is that the change from here to the bottom is probably yeast now adapting to this new environment. That sinking fast used to not be so advantageous, but now it is what the brewer is selecting for. And so you know, there might be a benefit to that. And so the other thing that I really like about this these small number of mutations that we found is that it really gives us some hypotheses about how we can have a cohort of traits kind of co-evolve in an environment like this. Like, how do you select directly on flavor in a big mixture? It's a little bit hard to think about. But if your flavor mutation that changes like there's alcohol content also causes a growth benefit or defect in, say, high osmolarity conditions, or is linked to a gene like the flow gene that causes a flocculation benefit, then for the price of one mutation, you can get multiple traits simultaneously. And so I think it's really interesting to think about how the architecture of linkage in the genome can affect the ability of yeast to adapt in particular ways and for traits to kind of co-evolve. Another thing to think about is, you know, mutants can be good or bad. Tom actually liked one of the evolved strains better. We took these strains that we made beer with the ancestor and 2 of the evolved strains and took them to HomebrewCon, which I have to say is one of the most fun conferences I've ever been to. And we did quote unquote sensory analysis, which is tasting beer and filling out a worksheet about it. And so some of the evolved strains actually had some flavor profiles that people liked. And so, you know, one thing that I've talked to Tom about is like, should we use a strain that already has this extra chromosome and the rearrangement for BAT1 and start his next batch of beer with that strain? Would it kind of quote unquote slow down evolution? Would they find new things to explore? I'm sure they would, but it would be interesting to see. So hopefully I can talk them into more science beer. We'll see. If you want to read more, we have a preprint out that you can go look up. And we're in the process of revising this right now, actually. So if any of you do read it and have any comments, I'd be interested in hearing them. And I just want to end with some unanswered questions. So one is that we really did a disservice to the pan genome here, where there are genes present in brewing yeast genomes that are are lacking in the lab strain, and we didn't pay quite enough attention to those. And so we're kind of going back and trying to look harder at these, maltose metabolism genes and flocculation genes. Yes, the lab strain of yeast can't eat maltose, which is ridiculous. But we've had to go and look at these genes in particular. The functional testing that we did of the BAT1 allele was in the lab strain. And so we really need to get better at these more complicated genomes, and so that we can test it in the context of the real genetic environment in which it's found. And then finally, I mentioned that this is with these Chico variety strains. And so we'd love to be able to do more of these experiments, but with different strain backgrounds, different brewing styles. I'm really excited that in the last year, we found a top cropper who's been willing to collect samples for me. So I'm really excited to see how that differs from the bottom collection. And I'm always on the lookout for lager yeast too. Those don't seem to be repitched quite as much. And finally, like various products, like I'm fascinated by the hard seltzers. Like the yeast are just put through a lot when they're making hard seltzer. So I haven't found anybody that does repitching for hard seltzer yet, but if you know anybody, I'd love to talk to them and take a look at their yeast, because I bet they are having to adapt. Okay, and then also, this is really a model for like all the different types of product, sourdough starter cereal transfer. Coffee beans and cocoa beans are fermented, and that is probably inoculated one to the next. And so we're starting to try to create some of these natural experiments that we can start to look at to understand how these have all adapted to these different environments. Okay, so I'm going to end just by I thanked everybody who contributed as I went along, I think. And this has been really fun to work with all these different brewers. I've learned a lot from them. Who cares more about yeast than a yeast geneticist? Somebody who uses yeast for a living. And so, and they have a great understanding of metabolism. Actually, one of the brewers caught an error in one of my papers that the reviewers at the journal didn't catch. And so, they've been really, really great and generous to work with. And we had a small grant from the American Society of Brewing Chemists for this, but it was mostly funded as broader impacts on an NSF grant, which was also the funding from a different NSF grant for the first part of the talk. So, and with that, I hope I have left a few minutes for questions. Thanks very much. Do any of the students have questions? I'll take student questions first. Oh, don't be shy. Yeah, sorry, I went a little bit fast too. Yeah, Chris.
Oh, I'll kick things off, but we can turn it back to the students if they have an idea. I think it's a two-part question. The first one is, did you know the Chico strain was tetraploid when you started, and is that why you picked it? Because some hill strains are
That's true.
Diploid, so I'm wondering, would you see a different landscape in diploid than I would your caveat?
Yeah, no.
I think it's interesting to think about what the difference between those.
A diploid ale strain would be awesome to work with. It would make our lives so much easier too. Yeah. We started with whatever Tom was using, and the Chico strains are really popular, and so it seemed like as good as anything to start with. Asking around, it's interesting. Brewers are not so adventurous with their yeast necessarily. Like Chico is used for everything. Like Tom sent me a graphic of all the different beers that he brewed with over the time course, and it's like, well, because it's, Seattle, it's like 4 IPAs and then a triple IPA and then, a porter and then a stout. And then the stouts actually are often branches because you don't want to repitch a dark beer into a light beer. And so he'll often like split it 3 ways, and one branch will go IPA or pale ale and the other branch will be a stout. And so it's like a good for everything kind of strain. So that's the other big uncontrolled variable here. The Elysian time course, they do a little bit more like one type of beer, a bunch of batches, just because they're a bigger operation. But yeah, I'd love to get somebody with other strains. So we've been, I have a few irons in the fire along those lines, but yeah, Chris has now moved on. He is a postdoc working in warm genetics now. And so I'm trying to find a new person who wants to work on beer yeast. So we'll see. And then your second question was caveat 3. Oh, that was that for that.
Yeah.
Okay. Yeah. Yeah.
I was going to ask if you noticed any differences in our DNA sequence throughout the experience.
That is a great question. And, we only did the short read sequencing. And so being in my department, I am wary of using short read sequencing for our DNA copy number, although I think you can probably make some inferences about that. But that it goes to kind of this caveat where we often ignored things that were complicated. And so we really should go and look at that more carefully. Yeah. And I'm sure that Bonnie Brewer's lab would run some pulse field gels for us. If that's a good question. Actually, I've never thought about that. Yeah. Other questions? Yeah.
The question about this bigger picture question, but with all of the studies that you're doing, especially looking at the genome evolution over time, do you ever learn things about genome organization? I think your lab is interested in other ways. Are there particular organization of genes on chromosomes or repetitive elements that could be hotspots, points that are next to genes that tend to vary a lot in these different environmental selections?
Yeah, that is really interesting. And I certainly think that the gene content of the subtelomeric regions is special. And like you don't find any essential genes there. And you find some genes that there's not a lot of, as you know, kind of functional clustering in the yeast genome, but there are some. And so like the mol genes in particular come in little groups of 3, and there are multiple cassettes of them. And many of those are close to the telomeres. And so a lot of transporters and other kind of metabolic bits and pieces are down there too. In addition to a bunch of genes, nobody knows what they really do much. So, and there are special repeats that we've found a few cases where we do think a transposon is next to a gene such that a recombination creates increased expression at that gene. And so, I think it's possible that and one thing that's been interesting is, we always are like, well, did we just make up a story about that because we see it? But with this new amount of natural variation that is available through, the sequencing projects like your lab and others have done, we've got all these strains we can look at and we can say, has natural variation explored this over and over and over again? And like with these brewing strains where we do see things we see over short term, we see in the diaspora of this yeast family. So we think that the short term stuff that we're seeing is actually possibly representative of what's going on over long time courses. And so it could be by comparing the things that we're seeing short term to the things that are differentiating different genomes that are collected from different times and spaces that we might start to see more patterns that are repeated use over evolutionary time, but also where in the lab we have now a hypothesis about what might be a selective environment to test that directly. I don't know, it's a really good question.
And so I have another quick one that you mentioned transportable elements. So are they very different? Are they moving along?
So we weren't able to detect any at high enough frequency to know, but the transposon calling, we often have to have higher read depth because you can only use these weirdo split reads, right? So we were able, we kind of, I think, took a look at it and weren't convinced of any that we saw. And so, but the clones from the first part, we had some really obvious ones that were real. So, but yeah, I think it'd be good to go and look more carefully at the brewing strains to see, like even for the different Chico variety strains, we didn't compare the transposon content of those, and those would be easy to find because they are clones that we sequenced. So. And there is such a huge variety in transposon content among strains. It's really fascinating. Where LTRs are and where transposons are highly correlated with Pol3 promoters. And so that is always, I think that is one element of how, and Pol3 promoters are also known to be sites of DNA breakage and kind of stalling and replication. So it has always been interesting to me that these sites of potential danger zones for DNA damage are right next to repetitive regions. And so our kind of transposons taking advantage of that, who knows, but kind of fun to think about. Other questions? All right, well, great, thanks so much. And thank you for the people who are online for joining us as well.